To say welcome to everybody. Thanks for joining. So the steering committee, which you'll learn about in a minute, has been working on putting together this joint series between GCOOS and SECOORA to bring you opportunities to better collaborate across two different networks, the Marine Biodiversity Observation Network and the Ocean Acidification Program. And so I'm going to introduce Dr. Kim Yates, who wears many hats. She works for the U.S. Geological Survey. She is on the board of directors for the Gulf of Mexico Coastal Ocean Observing System, and she is chair of the GCAN network, which is the Gulf of Mexico Coastal Acidification Network. And so I'm going to hand it off to you, Kim. And then at the end of the presentations, this is the first in a series of 5 webinars. Jen Vreeland, who's also has been very instrumental in organizing the webinar series, will outline what the next steps are going to be and where folks can go to register. Thanks, Jen. Where folks can go to register and learn about the next webinars coming up in the series. So all yours, Kim. Thanks.
Thanks, Chris. Welcome, everyone. We're happy to have all of you today for our first Gulf of Mexico Coastal Ocean Observing System and Southeast Coastal and Ocean Regional Association Collaborative Spring Webinar Series, titled Building Synergy Across the U.S. Marine Biodiversity Observation Network and Ocean Acidification Networks. So as Chris was saying, this year GCOOS and SECOORA teamed up to develop a webinar series focused on establishing collaborative opportunities among MBON, the Gulf of Mexico Coastal Acidification Network, and the Southeast Ocean Coastal Acidification Network. And as she mentioned, this is our first webinar in a series of 5 to be hosted over the next few months. And today we'll be kicking off our series with overview presentations on MBON and NOAA AOML ocean acidification program activities in the Gulf of Mexico and Southeast regions. And I want to thank our co-organizers for developing the series as a way to help find intersections between MBON and OA network activities that we hope will foster new collaborations to integrate research and monitoring across these fields of science. And so thanks go to Dr. Chris Simonella from GCOOS, Jennifer Vreeland Dawson from GCOOS-GCAN, Dr. Emily Hall from Mote Marine Lab and SOCAN, Jennifer Dorton from SECOORA, and Emily Noakes from SECOORA. So before we get started on our presentations, we'd like to give you a brief overview of for our webinar series. Our goal for this series is to advance science in support of resource management and the blue economy by strengthening collaborations across MBON and the regional acidification networks, GCAN and SOCAN. And we've designed this series to help improve understanding of the state of the science, gaps and needs for marine biodiversity and ocean acidification research and monitoring in the southeastern Gulf of Mexico, and to identify opportunities to bridge and integrate science across marine biodiversity and ocean acidification research and monitoring, and to synthesize lessons learned across MBON and OA activities to inform new partnerships and collaborative research and monitoring opportunities across our regions. We anticipate that this series could provide the foundation for a variety of outcomes, including potentially a white paper or manuscript summarizing our key findings, lessons learned, gaps, and needs for research and monitoring, for improving collaborative research and monitoring activities across our regions, and potentially for a workshop informed by these webinars to identify priorities for collaborative research and monitoring activities in our regions. The format for all of the webinars in our series will include a 20-minute presentation on OA activities, followed by a 20-minute presentation on MBON activities, and then we'll have 20 minutes for questions and discussion for the presenters and the participants. Over the course of the next few months, our speakers are going to be focusing on discussing the state of the science for OA and MBON, major information gaps in their specific areas of work, the benefits of communicating and collaborating across our networks, and challenges to collaborating across the networks. So today we're going to begin our series with a presentation on OA activities from Dr. Anna Palacio. Anna is an assistant scientist at the University of Miami Cooperative Institute for Marine and Atmospheric Studies, and she's also an affiliate with the NOAA Atlantic Oceanographic and Meteorological Laboratory Ocean Chemistry and Ecosystem Division. She's going to be giving a talk on AOML Coral Program's contribution to OA monitoring and research. And that will be followed by a presentation from Gabrielle Canonico titled The Evolution and Status of the U.S. Marine Biodiversity Observation Network. Gabrielle is a manager for the U.S. IOOS Marine Life Program and the U.S. Marine Biodiversity Observation Network. And she's also co-chair of the Global Ocean Observing System Biology and Ecosystem Panel. And Gabrielle asked me to post this link at the bottom. This is a link for the Ocean Acidification Research for Sustainability white paper that was just published in 2023. And this paper outlines an international strategy for integrating and communicating ocean acidification and marine biodiversity science to provide timely information on the impact of OA on marine life that informs policy and management actions that we can use to inform our regional discussions and future planning for this group. So we'll also post this link in the chat. And I just want to open the floor for if there are any other comments from our organizers while we're transitioning to Anna's first presentation. And I'm going to stop sharing my screen. 1 second.
I'm going to start sharing if there are no further comments. Does it look good?
It's perfect. Thank you.
Awesome. Thank you. Yeah, welcome everyone. I'm super excited to be here today and share some of the ocean acidification research that we do. My name is Ana Palacio. I am a SEMA scientist working at the AOML's Coral program. And a lot of the work that I'm gonna show here, it's also an effort pushed by my colleagues, Nicole and Ian. So I would like to also thank them for a lot of the work that they have done that I'm about to present. I'm gonna try to divide my presentation in 3 big topics. I'm gonna start talking a little bit about the ocean acidification monitoring that we do as part of the National Coral Reef Monitoring Program, or NCRAMP. Then I will briefly touch base on our ex situ ocean acidification research capabilities at the Experimental Reef Lab. And then I'm going to finish mentioning some of our ongoing ocean acidification research and collaborations. First, I want to mention that the National Coral Reef Monitoring Program is a multi-agency effort. It was established in 2013 by the NOAA Coral Reef Conservation Program. So there are a lot of people involved in the monitoring. But AOML's contribution to NCRAMP is concentrated in climate, including ocean acidification, with a strong presence in the Caribbean and the Atlantic region. As part of our monitoring efforts, we visit these locations that I'm showing in the map, and we come and visit these places every 2 years to collect samples and data and to deploy and retrieve instruments. However, our sampling efforts are like switched in between the 2 years. We first visit the flower garden banks, the dry tortugas, and the South Florida places in 1 year. And then we visit the U.S. Virgin Islands and Puerto Rico in the following year. During these expeditions, we like to target a variety of data types. This is to try to capture both spatial and temporary variability in carbonate chemistry. First, in our expeditions, we collect 50 bottle samples in each one of these locations, and those are analyzed for TA, DIC, and pH, and we use that data to calculate the saturation state and PCO2. These samples are collected in random locations near the reefs, but we also target diel cycles in the reefs, and for this, we collect samples every 3 hours for a period of 72 hours in the reefs. And in order to do so, we use these devices that you can see here in the picture. These are called the submerged automated samplers, and they allow us to collect samples even at night. And I also want to mention that we target temporarily rich data with the map CO2 buoy that is located at Chica Rocks here in the upper Florida Keys. This buoy is run in collaboration with the NOAA Pacific Marine Environmental Laboratory, and it's located in a very important spot for coral reefs. Chica Rock is one of the most resilient reefs in the Florida Keys. It has been resistant to bleaching and disease, so it's quite of a site of interest for us. This is some of the snapshot of the data collected by the buoy. It's hosted between other places in the SECOORA data portal, and as you can see, it provides high resolution carbonic chemistry. It monitors ocean surface as well as air PCO2 and pH, along with temperature and salinity. I want to kind of zoom into the data that we collect in the Florida Reef. Our Florida Keys data series for the bottle samples is a little bit more comprehensive because in this location, our sampling effort is a collaboration between NCRAMP and the South Florida Ecosystem and Restoration Research Cruises. So as part of these cruises, 38 stations along and across the Florida Keys are sampled. These stations are divided between or cover 4 subregions in the Florida Keys: Biscayne, the Upper Keys, Middle Keys, and Lower Keys. And as you can see, we run this, we collect this data on some sort of like transect that allows us to capture different zones of the reef, such as the inshore, mid-channel, and offshore areas of the reef. The data has been collected since 2011 to present, and we have kind of every 2 months samples that are being collected starting in 2015. Before, the data collection wasn't as frequent, but this is one of the largest synthesis of carbonic chemistry for the Florida Keys to date. And I'm gonna kind of like show a little bit of the richness of the result that this data has provided us to understand the region. If you want more details, you can check our paper here that was recently published. But I want to highlight here that the data allow us to identify differences in seasonality across the different regions, across the different zones of the reef. So we can see like high seasonal variability in the inshore reefs, and this variability decline as you move offshore, which informs us about the condition that the corals and the coral reefs are experiencing in different areas of the Florida Keys. But we can also see in the different colors we have the regions inside the keys. We can see that some regions, such as the ones in green here, which is the Upper Keys, experience higher pH and aragonite saturation state values compared to other regions, such as the Lower Keys and purple in the bottom. Showing also as important information for coral reef conservation and restoration. We need to understand what are the conditions that these reefs are gonna experience. And additionally, we were able to estimate interannual trends with this database, and I'm just gonna zoom in into the pH levels to show that we were able to detect faster changes or like faster acidification in the offshore reefs when we compare them with the inshore reefs, which is it's not necessarily the case for most reef ecosystems, where like inshore reefs are commonly exposed to exacerbated ocean acidification, and we're seeing different patterns for the Florida Keys. So a lot of interesting data. I just want to give you that little piece for now. We are a coral lab, so coral reefs are our target ecosystem. And part of, Seems like I cannot play this. Oh, yeah, it's playing now. So a lot of the oceanographic work that we do tries to understand how oceanographic affects a future reef persistent, or is going to affect future reef persistent. And reef persistence is determined by the balance between reef growth and erosion. So we studied these questions using calcification accretion units that we do in collaboration with the NCRAMP Pacific team, as well as with bioerosion monitoring units, which is data that I'm zooming in here and the data show in the video. So we deploy these units in the reef and then collect them 3 years later to understand accretion and bioerosion in the different NCRAMP locations. Another type of data that we collect are carbonate budgets. Carbonate budgets is a census-based approach that uses the abundance of key carbonate producing, such as corals, and eroding taxa, such as sponges or parrotfish, to estimate the net rate of carbonate production in a reef. And here, for example, in the right panel, I'm showing the data for the East Flower Garden banks that we collected in 2015, 19, and 22. And here the big panel is the overall carbonate budget, which is showing as positive values, which means this reef is growing. And in the smaller panels, we have like the different processes that contribute to this overall carbonate budget. But unfortunately, this growing reef is not necessarily the case for most of the locations in the Caribbean. In this paper, for example, you can scan it here for more details, but in this paper, we found that at least 70% of the reefs in Florida are in a net erosional state. These are shown in the red and yellow colors with negative numbers for the carbonate budget, and you can see that the positive green values are very scary in this map. So yeah, it's not like a happy picture for all the locations that we study. Finally, in the field, we also collect landscape mosaics data, which are used to quantify the benthic community and monitor changes in coral cover over time. To make these mosaics, we collect thousands of pictures underwater, and these are then stitched digitally to make a high resolution image of the reef that is some sort of like a snapshot of how the location look at the sampling time. And this data has helped us monitor both like coral cover, but also the effects of heat waves. And here I'm showing data that we collected the past summer, and you can see how all the hard corals are bleached after the heat wave, and the sea fans that were alive here are completely dead a month after we collected this first data in June 30th. So we use this to just monitor and report what's happening in the reef over time. To kind of like close my National Coral Reef monitoring part of the talk, I want to mention that besides the scientific publications, NCRAMP results are summarized in a status reports that are available to the public. And we also put together short climate monitoring briefs that we complete after each field campaign. These briefs describe the type of data that is collected under each expedition, the instruments that were retrieved or deployed. So it's also some sort of like information that is available for people to know what kind of data we have. Here, I'm gonna completely switch gears from the field to the experimental reef lab. to touch a little bit on the ocean acidification research that we are conducting ex situ. So the experimental reef lab is a state-of-the-art lab. It has 16 independent tanks. The lab was built to be able to mimic ocean acidification and warming conditions to study the effects of climate change on reef organisms. So we have the capability of manipulating pH conditions, for example, with great accuracy and precision. And you can see here in this right panels, we have the capability of reaching different treatment levels, but also incorporating different amplitudes and dial variation in pH, for example, which is so much more informative of the natural conditions that we experience in the reef. So this lab has been instrumental on testing the effects of ocean acidification, as well as variability into the diurnal cycles in pH, and the calcification of corals, bioerosion rates of sponges, as well as dissolution of coral rubble. So there is a lot of possibilities and capabilities that we could implement to learn more about specific organisms' response to ocean acidification. We also recently developed the sequential treatment application robot system. This publication became available like a week ago. So again, if you want to learn more, scan this Q code. The START system allows us to incorporate it's a high replication system where we have a lot of independent basals in each tank and allow us to simultaneously test stressors such as ocean acidification, and we can see interactions with, for example, nutrients, warming, disease, etc., and see how additional stressors work together with ocean acidification. But it also allows us to test a variety of ocean acidification levels. We can move away from testing just like low, medium, high treatments and incorporate tens of different levels on those treatments. Now I'm gonna, just to close, touch base about some of the ongoing projects and collaborations that we have. Excuse me. This first one is Project Fresca, or Florida Region, excuse me, Florida Regional Ecosystem Stressor Collaborative Assessment. Project Fresca is also a multi-agency effort. It's led by AOML and the University of Miami. And our main goal is to understand how marine ecosystems will respond to the combined effects of ocean acidification, warming, hypoxia, harmful algal blooms, and nutrient pollution. The project is big, so it's divided in between big modules. But some of the work include to characterize the distribution of these stressors I just mentioned across Florida, and to test individual species response to those stressors to determine thresholds where their performance just switch by being exposed to OA and hypoxia, for example. And then we want to incorporate all this information into modeling efforts to understand the potential future climate scenarios and how the ecosystem will respond to those future scenarios. And future scenarios include both climate change scenarios, but also potential restoration scenarios. We want to understand if we restore certain ecosystems, how we can change the ecosystem dynamics and resilience to the stressors. Finally, I want to mention our collaboration with Mission Iconic Reefs project. For the Mission Iconic Reefs, we want to support or incorporate comprehensive climate monitoring at the Mission Iconic Reef sites. This includes spatiotemporal characterization of ocean acidification and warming. So we are specifically targeting the fourth northernmost reefs in the Mission Iconic Reef project, and in those reefs we are deploying pH and temperature sensors, as well as collecting bottle samples to understand carbon chemistry, and we hope to support the work that Mission Iconic Reef is doing in these sites. That's all I had to share with you today. I would like to thank all the AML Coral Program team. All the work I show is a joint effort between our lab as well as our collaborators. I think we're taking questions.
Thank you, Anna. I think we're going to right now, we're going to hold questions until we're done, and then we're going to have a good long section of time to go back to questions. I'm going to hand it off back to Kim, who's going to introduce our next speaker, Gabrielle Canonico from the US IOOS office. So Kim, all yours.
Thanks, and can you help Gabrielle get transition with her screen? And so again, we want to welcome and thank Gabrielle Canonico for her presentation today. The title is The Evolution and Status of the U.S. Marine Biodiversity Observation Network. And with that, Gabrielle, I will let you take it away.
Okay, thank you. And are you able to see my slides?
Yes, we are, and we can hear you clearly.
Wonderful, thanks. Yeah, so I'm really happy to be part of this conversation, and MBON has for a long time been engaging with ocean acidification researchers and partners to try and develop routines for how we can better align biodiversity observations with those of ocean acidification parameters, or to engage the OA community that's taking biological measurements to contribute those data for broader biodiversity understanding. So there's a lot of opportunity for kind of collaboration and work together in this space. My talk is going to be more of a programmatic one, less of an interesting science one, so that's a little different from the previous talk. But I did want to mention that MBON has an active project, and I'll talk about that in a few minutes, but in South Florida that has links to the Gulf of Mexico also, and is routinely a part of those bi-monthly cruises. And I'm assuming they're the same ones that Anna was talking about. So already there are mechanisms in place for us to think about better leveraging. So MBON is really kind of in the center of a global conversation about how we can better, more effectively answer biodiversity questions that are looming large and very important for society in the context of the Sustainable Development Goals, conversations at a high level about a sustainable ocean economy, looking at offshore development and the need to really understand what impact we're having on living resources that humans are so reliant on. And so I'm going to start just by sort of introducing the global context for this, and I think I'll end on a global note as well with a little bit about US MBON in the middle of the talk. But really, MBON is part of a large partnership that's trying to understand what changes are occurring with regard to marine life, what are the impacts of those changes on human society, on ecosystem health, and what we need to understand about potential for recovery from those changes. And so this is a bit of a timeline that illustrates sort of how we get to an MBON. The timeline starts with GCOOS and the establishment of expert panels on climate, biogeochemistry, and eventually over the course of the timeline also on biology and ecosystems. But really, MBON was born out of the Census of Marine Life, which was a 10-year effort to take a snapshot of marine life. It ended in 2010. In the US, we took to heart the recommendation or the acknowledgement as part of the census that there really is no systematic way to document and understand marine life and the changes that are impacting it. And that this was something of critical importance that US agencies were interested to support. So I had put a NOP logo on my opening slide. NOP is the National Oceanographic Partnership Program, and it's really an initiative that brings together federal agencies with academic, other non-government sectors, industry, into conversations about priorities of mutual interest and looking for leveraging opportunities. So NOP has long been, has always been part of the driving kind of initiative behind MBON, very much an interagency priority to understand how can we establish an observing capability for biodiversity that aligns with global needs and global emerging standards. Most recently, the MBON team the MBON team had reached out broadly across some of our global partners and proposed a UN Ocean Decade program, which was endorsed. It's the Marine Life 2030 program, and I co-lead that with Frank Miller-Carter from University of South Florida, who I think would have loved to have been in this conversation today, but he's in Barcelona for the Decade Conference. So Marine Life 2030 broadens the reach of MBON, expands our global partnerships. A number of the key partners are listed on the slide, including a biomolecular observing network, a deep ocean observing one, a couple of networks very focused on fisheries, and so on. So that's a really interesting opportunity to expand our reach. But I said I'd circle back to some of the US interagency interests. And so MBON was launched as a series of demonstration projects in 2014. Between 2014 and today, we have invested over $30 million from NOAA, NASA, BOEM, Office of Naval Research. Initially, Shell Oil was at the table with an Arctic project that launched in 2014. And so this is just a quick list of where we've supported some activities. I'll show you a picture on the next page of some of the currently active ones. In FY22, we launched 5 new projects, and I listed this really long title because I just wanted to highlight that MBON is always looking for collaborations across networks, not just with OA, but in strengthening linkages to animal telemetry observations, and all with a focus on how we bring that information into applications for ecosystem and human needs. In FY24, we are going to be announcing 5 additional projects supported with Inflation Reduction Act funds, but those are forthcoming. So this is a really busy picture, but I just wanted to highlight the projects highlighted in yellow are the ones that are currently active. I said we have 5. We have 5 US MBON projects. The 6th one in the lower right-hand corner that's highlighted is the MBON Pole-to-Pole initiative in the Americas region, which is led by Enrique Montes from AOML, who has long been an MBON partner. And this network is making tremendous strides in terms of global coordination in that region and building on standards and protocols for nearshore, rocky, intertidal, sandy beach observations of biodiversity, and looking at opportunities to expand those more broadly in the Atlantic Basin. So that's an evolving initiative. So the US MBON is, as I've alluded to, part of a global partnership. It's also what is considered a thematic node under the group on Earth observations. And so US MBON is one of a series of regional MBONs. There's also an Asia Pacific MBON. There's an emerging MBON in the EU. There's the Americas initiative I just mentioned. But all of these are really thinking about how we can better operationalize the collection and sharing of marine life observations, leveraging existing activities, creating new ones to fill the gaps where we need to. All of this, because it's such a significant undertaking, really relies on establishing networks of different types of partners, including engaging members of the public to share information and ways of documenting how marine life resources and their availability is changing. We've also long had an interest in bringing biodiversity observations into the ecosystem forecasting and modeling discourse, where there are some gaps in that information availability for forecasting. And a real priority for us has been advancing interoperability of methods on the ground, data management and data sharing approaches, and also applications, so that to the extent possible, we can create initiatives that can sort of self-perpetuate and build on themselves. In some cases, we have projects working in different regions, but with similar partners. Are there ways that we can leverage the work that we're doing with those partners? And then contributing to global progress on expanded marine life observing and data sharing is a priority for many of us in the MBON activity. And that includes the Ocean Decade as well as other global initiatives. So just a short list, this is not comprehensive, but some of the successes of US MBON over the years include operational production by NOAA Coast Watch of products called MBON Seascapes, which are essentially maps of dynamic habitats that are derived from remote sensing and other types of data, and provide an opportunity for us to integrate a look at changing habitats with information that we collect about species, and to be able to say something about how distributions, presence, absence, etc., are changing.
We lost your presentation. I'm sorry.
Lost it.
I don't know if this happened for everyone, but I cannot see your slides anymore. I'm
No.
Here you go.
Is it back? Yeah, it blinked, so obviously that's what happened. Okay, thank you. Okay, so MBON successes. So Coast Watch, for US MBON, it's always been a priority to figure out how we can best engage with US IOOS regional associations as really backbone observing infrastructure and stakeholder engagement infrastructure, because those partnerships are going to be key to our long-term sustainability in the US. MBON partners have been instrumental in advancing documentation about eDNA methods for biodiversity understanding and looking at opportunities for application in that regard, but also doing this with other technologies, imagery, passive acoustics, integration of telemetry, and multiple different types of biodiversity observations to understand hotspots. So technology has been a focus since the outset. I mentioned a couple times promoting common data approaches, providing science for real on-the-ground use and application, building student and workforce base and understanding about biodiversity observation methods and standard approaches for data and information sharing has been a priority. And I've said a few times, the global engagement is really foundational to ensuring that we're developing common approaches, not just for the US, but that have much broader relevance. And those things highlighted in blue, I'm just going to speak a little bit more about in the next slides. This is really busy, but building a biodiversity data community has been a priority. So we really recognize that to the extent possible, when we're managing biological data in standard ways, based on standard formats and contributing to open repositories, we're making those data as accessible as possible for multiple different uses. So IOOS with the Ocean Biodiversity Information System, with some of our other GCOOS regional alliances, with MBON, the Ocean Tracking Network, we've hosted jointly a number of biological data mobilization workshops to build communities around aligning data with the Darwin Core Standard for biodiversity information. a number of our colleagues are involved in the Earth Systems Information Partnership, and there's a little box titled Biological Observation Data Standardization is something of a primer for people who are involved in biological data management. That's available on the ESIP website that came out of that group. So there's a number of efforts to try and really build awareness about that need for and mechanisms for standardizing biological data. I think there's a lot of opportunity for our MBON OA partnership in this space. So I hope it can be a feature of our conversation going forward. I also wanted to flag a couple types of products that MBON has been instrumental in creating. One is with the Office of National Marine Sanctuaries, working together with sanctuaries, with IOOS, with other partners to develop these web-enabled platforms for information that feeds sanctuary condition reports. I've provided some web links here because you might find it interesting to browse these a little bit. It's really providing, it's building bridges between the science and the observing work that we do, sort of through translation to make that data and information relevant and more accessible for management. And we know that we're providing that service because it took us a while to sort of evolve the processes to develop these infographics and to develop tools for digging into data that were friendly for use by managers. So a lot of one-on-one work with the management community on these products. I did want to flag in that last bullet, so Sanctuary Watch products tend to be regional because they're focused on a site. So you can see Channel Islands, Florida Keys, IEA, and Olympic Coast are some that we've got on the website now. But we also have thematic ones, and the first of those was called, with supporting the Sanctuary Soundscape Monitoring Project. I think this is an interesting model, and in fact, I've heard from Sanctuary's partners that there's interest in building out a suite of products for Sanctuary Watch that provide information about OA impacts on biodiversity. So just present that as another potential opportunity for our dialogue going forward. And then I wanted to flag the early alert dashboards that were developed, starting with Flower Garden Banks following a coral die-off event a number of years back, but expanding now to Florida Keys. The interest in providing sort of dashboards that give access that can manager into an event happening in their area of responsibility, that they can then kind of look at what some of the trends are in whatever data they're interested in that helps them to understand the event. Provided a link there too in case folks want to dig in. So I'm circling back to the global effort here. MBON is an interesting resource because it's common practices that are really actively being discussed in the global community now, and which US IOOS, for example, has expressed a priority to support. So the Global Ocean Observing System is identifying essential ocean variables or has identified to prioritize the observations we take as a global system. Some of those are biology and ecosystem variables. MBON is a network bringing together the biological observations community to advance implementation or deployment or collection of those observations and support research and development that's relevant for biodiversity observing. Ocean Biodiversity Information System is a data partner focused on promulgating promoting data standards, including Darwin Core, and building tools for data visualization and analysis, and linking to larger global data repositories as well. So it's a very carefully thought out schematic, and I think there's opportunity, as we think about the OA community and where they provide input. For example, on the biogeochemistry side of GCOOS and prioritizing observations we collect for biogeochemistry. That's an analogous process to the one we've developed for prioritizing biological observations. And then the Ocean Acidification Research for Sustainability is a UN Ocean Decade program that several of us have been giving input to. In particular, Frank and I, along with Sam DuPont, have been championing an outcome focused on biological impacts from ocean acidification. And so that's why I would encourage this group to look at the white paper that was developed with a focus on biological impacts, although the white paper resource that OARS provides at this link here goes broader than biology and is really an interesting resource to look at. So there are some recommendations in the white paper, including around building communities among the OA research effort, building communities within that kind of OA focus that support the standard data sharing and data management processes for biological data that are co-located with those OA data. And I think there's some really interesting opportunity there. So please take a look at that paper. And I feel like I flew through this because I'm also super keen on dialogue and questions. So I'm going to stop sharing and stop talking. Thank you so much.
Thank you so much, Gabrielle. So both you and Anna did a fantastic job outlining the foundation for OA and MBON discussion. We're going to go ahead. There's not that many of us on the call. There's about 38 people. So we have two options for you. You can raise your hand and just ask your questions, or you can write it in the chat. And Jen and I will be monitoring both the chat and the hand raising. So if anybody would like to get us started, I will refrain myself because I have a whole bunch of questions from that interesting dialogue. I guess while we're waiting to get started, my first question, Anna, I was curious about the STAR system. That's fantastic. I mean, in all the field work that we've ever done, trying to control various parameters has always been a challenge. And so that ability is fantastic. Are you looking right now at single species, or like from the marine biodiversity side of things, thinking about ecosystem function, would you be able to add species of fish or crustaceans and look at the interplay between organisms under those controlled settings?
Yeah, we're looking forward into incorporating a massive amount of like species in our coming up experiments for testing. Right now, we are modifying the size of the individual containers or individual like experimental chambers that the STAR system hosts. So we can definitely expand and change sizes to provide the conditions necessarily to test different organisms. For the Frisca project, we are going to test, besides corals, we're going to incorporate some of the sponges from the Caribbean, as well as seagrass and sargassum, to test the responses to ocean acidification, hypoxia, and temperature. So we're definitely like looking into testing a variety of organisms. I think I don't know about fish. That may be that may take a little bit of extra work to be sure that we could provide like a good environment to test fish. But if people have ideas, we're happy to hear them and try to make it work.
Thank you. Yeah, I could see there being a lot of interest across the MBON network for that type of a testing system. Thanks.
So
Kim,
thanks, Anna. And we have a hand raised. I believe it's Kim Yates.
Thanks, Anna and Gabrielle, for fantastic presentations. I actually have two questions, and my first one is for Gabrielle. I'm really interested in the Sanctuary Watch products, and just want to throw it out there that I think we would be really interested in trying to move something like that forward for at least the Gulf of Mexico region. And I think Jinping was on the call, and he's been doing a lot of the Flower Gardens National Marine Sanctuary work, so we may try to get him to jump on board for something like that as well. So I guess my question would be, how do we start engaging with you for moving towards a product like that for our region, or for the
I think
for flower?
sorry.
Go ahead.
Yeah, no, I think let's just find time for an initial conversation. And maybe if you take a look at the products that are available on the website now and sort of think about what makes sense for your region, or what doesn't, that would be a little bit of good background to bring into a conversation. We are sort of leveraging the approach from Sanctuary Watch for some marine mammal health related infographics, working with GCOOS now. So I think there might be something to learn from that process, which is just getting off the ground, which is not necessarily focused on a sanctuary site, but is focused on an issue of concern in the region. So I think let's talk.
Yeah, that would be great. We'll reach out later and try to set up a conversation with you about that. And then Anna, I had a question about your experimental reef lab activities that you have going on there, and I heard that you're looking at dissolution of coral rubble. And I wonder if you all are already or thinking about also looking at reef sands and sediments in that system.
Yeah, so when we were planning this experiment, sands and sediments were the first thing that we wanted to target. My colleague that was leading the experiment ended up going for rubble to understand the dynamics in rubble. But dissolution of reef sediments is one of the biggest questions and gaps that we need to start addressing. So I think this is definitely something that we want to do in the future.
Great. We'd like to reach out to you from USGS when you all start planning that. That's an area that we're also working on in relation to our C4 elevation change projections and modeling for the Florida Keys Reef Tract. So that's an important variable that we're trying to work into our projection models as well. So I'll ping you about that behind the scenes as well.
Absolutely, yeah.
Great. Thank you very much to both of you.
And Jinping commented that he's happy to contribute data to that effort as well. So one area that MBON is keenly interested in, this year will be the 10-year anniversary of MBON projects being funded, and we're looking into more synthesis type projects, and I'm hearing a lot of great work that AOML and the OA network has been conducting, and I hope people will start thinking in terms of what some of those synthesis projects might look like, or products. So I'm especially interested, Anna, you said it's kind of unprecedented, the level of resolution on the pH that you're going to be able to be testing. Is that just in a controlled environmental setting, a controlled setting, or is that going to be in situ observations as well? Because I can see that being really valuable to refine some of the seascapes that are already out there.
So in the part of the experimental reef lab, we talk about how we can manipulate pH to a very high accuracy and precision. We do have some field monitoring of pH that we are doing for the SOFAR buoys for the mission iconic reefs. I would like maybe if Ian is in the call to kind of like talk a little bit more about the future in the field monitoring plans that we may have.
Yeah, thank you. So I think one of the things we've been trying to wrap our brains around is how do we get high temporal resolution data in like near real time, analogous to our MAP CO2 buoys, but don't incur the massive costs of MAP CO2 buoys. And so exactly like Anna was saying, we are interfacing in some of our instrumentation, particularly the SAMI pH instruments, with SOFAR buoys, which are these, I don't know if you guys are familiar with them, there's these cheaper kind of buoys that have cellular and some satellite relay capabilities. And so the idea is to, again, just to have some real-time feedback on our pH monitoring. Once we have just started to implement these communication protocols successfully. I'm really happy about that. Go Taylor. Moving forward with it. In terms of manipulation in the field, we have not been able to accomplish anything like Kim has done, with our, large tenting aspects. But we are, yeah, that's where we're at. I hope that answers your question.
Thanks. And I'm seeing some really positive comments in the chat box looking at collaborations that are already seem like they're going to be underway based on some of the dialogue. So that's really exciting to see. That's exactly what the intent of these sessions are. Jen, are you seeing any other questions in the chat or from hands?
I am not seeing any hands, and I don't see any new questions in the chat box.
Anybody else, any other thoughts or comments or questions for our panelists? We also have other experts from both SOCAN and GCAN on the call. So if there are topics or questions relevant to those, feel free to ask. We have other folks engaged in this research as well. And Gabrielle mentioned Enrique was working on the pole-to-pole for more international work. He will also be presenting during the GCOOS members meeting that will be taking place in Galveston in May. And everybody's welcome to join that. We'll be sending out some information as well on the opportunity to participate in that meeting. I'm looking just to see any more comments here. I'm not seeing any. We're coming up on the hour. Jen, would you like to give folks a little taste of what's to come as far as the rest of the series?
Well, we're still working on confirmations for presenters, but as soon as we have these confirmations, I'll be sending out to all of our registrants for our first webinar insight and information on how to register for our remaining 4 webinars to come. So stay tuned, and I'll be sure to reach out to everyone with that information.
Great, thanks. And our next one will be April 30th. We'll be hearing from Luke Thompson and from, who else are we hearing from, Jen? I'm blanking right now. We're going to hear from Luke Thompson and Neil Hammerschlag on shark research and animal tracking and telemetry, and from Jennifer Mixis-Olds from the University of New Hampshire on some passive acoustics. So it'll be more on the living marine resource side of acoustics. And I think with that, we will thank our presenters. Thanks to Anna and to Gabrielle for laying the foundation for us. Thank you very much to Kim for hosting us today and for serving as our moderator, and to all of our planning team members who helped make this happen today, and for all of you for joining us. Jen, we'll be posting, this session is being recorded, so these will be available following the webinars if you'd like to review them and have access to some of the amazing graphics and stuff that were provided today by our speakers.